Hello to all. What I want to guide you through this time is another mini lecture on the chapter on energy from your book. And I wanted to start out with a couple of laws. I know it sounds like physics class here, but these couple of laws really give you an idea of energy, why we're thinking of energy, how do you get energy, where does it come from? So the first law is going to tell us a little bit about that. The total amount of energy in the universe is constant. Constant. So if we are taking it, we're taking it from some other source that was using it or had it. So I'm telling you in this slide, given that our bodies cannot create energy, our bodies must import, that's a nice way of saying it, from the universe. We're taking it from somewhere else. The second law, I'm showing you a picture here of myself, a picture of a tidy room and a very messy room. This is a kid's room, little kid's bedroom. And this is actually a law saying that natural, spontaneous processes move from a state of order, the room on the right, to a condition of disorder or randomness known as entropy. So what does that matter? Well, your body or any living thing has to maintain that order of acquiring energy, keeping your cells alive. And if you are unable to do that, it's known as entropy. So actually illness, sickness, being ill is a little bit of entropy. It's disorder when the system isn't working well. Okay? So just remember, things that are natural processes will move from this order. So where it's a total input of energy all the time in order to keep it tidy. So the room on the right is energy inserted into this room every day for it not to turn into the one next door. So ultimately, life remains ordered, right? Mostly it has an order to it because of the sun. It's constantly supplying that energy to Earth. But we do know that entropy of the universe as a whole, including the sun, is always increasing, but we'll leave that to the physics guys. This is just a picture of a pine cone, but it shows one that has had an input of energy and it's been taken care of to a one that is moving towards entropy. So not doing well. Okay, and I will let you read more about that in your text and look at some of the other videos that I've supplied. What I do want to talk about though is ATP, the molecule of ATP. If you're not familiar with this molecule, you do have to learn about it. And this is what it looks like when written out. So you have, if it's adenosine triphosphate, it's because it has 3 phosphate groups. Okay, so here you can see the phosphorus with an oxygen, 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 oxygen. This is a phosphate. It also has a double bond. Phosphate number 2, phosphate number 3, triphosphate. These triphosphates are attached onto a sugar, ribose, and a base with nitrogen in it, with protein here. So this is adenine. And adenine together with a ribose is going to make adenosine. Hence the name adenosine triphosphate. Okay, super important molecule. This is the molecule that keeps you ticking. It keeps you going and not turning you into the crumpled up pine cone. Kind of. So how is it that we use that molecule? We use that molecule as our source of energy. The classic analogy is you have a savings account, which is your fat. And then you have a checking account or cash that you're going to be paying out when you need something. You need to pay something. And that is what ATP does. We don't store a lot of it. No, we use it for that energy that we're using for absolutely everything that we do. So how does that work? I'm reading this to you now, removing the N-most phosphate group by hydrolysis releases the potential energy stored in ATP. The cell uses this energy to do work. What's that mean? This was that original molecule I showed you. Okay, hydrolysis means that I'm going to lyse water. Lysis means to break. So break water and put some of that in. And I'm going to take away one of the phosphates. So in this diagram, it's being shown with an arrow, taking it away. Take away that phosphate. It's that phosphate that is going to attach onto the molecule that's doing work. Whether it's you moving a muscle, let's think of it as that. You're moving a muscle. In order for that muscle to move, it needs that little phosphate. That phosphate needs to attach. So it's the use of that P. And you're left behind with another molecule that you really can't use for energy, and it's only got 2 phosphates, so it's called adenosine diphosphate, ADP. But you got some energy out of it. So how do you restore that? You can recycle the ADP, attach on another phosphate, and you have another molecule ready to donate a phosphate to you when you need to do something like contract a muscle. Let's stay with that example. This slide is telling us ATP provides the energy to build large molecules out of small subunits. So when we were looking at large carbohydrates, they were joined one to another in these long chains. Well, that doesn't happen without the input of energy. So ATP also, it needs to be present. What part of it? That particular phosphate. That one phosphate is going to be part of that molecule. You need that input of that phosphate to manufacture these long chains. Okay, so very important, this molecule of ATP. Also, crazy is how much we store of it. We store enough for about 2 minutes. That's nothing. So if you stopped making ATP right now, you still would have 2 minutes left. So it's a good thing that we have fat, because that's what's going to generate some of that ATP. So here I'm telling you more about that. It's a very unstable molecule. It's not something you store, and you store 2 minutes worth. A human cell uses about 2 billion ATP molecules per minute, because it's not just muscle, it's everything. Every time you have a thought, every time you think something, there's ATPs needed to be used in that reaction. How do we make it? That's the next question that comes up. How do we make ATP? Well, that is made in the mitochondria. And this is a picture of a mitochondria with all these little, it looks like a maze here. And this entire lining of this maze is full of enzymes. Enzymes that are making this reaction happen. So why don't we look at this backwards? If we want to make ATP, we come backwards here. What we needed were the raw materials. And the raw materials to make that are glucose. You need glucose. You need to be eating. The second one is oxygen. You need to be breathing. And then you need that leftover molecule of ADP that we talked about, because you're going to tag that phosphate onto that ADP. Once you do that, your outcome here is going to be the manufacturing of that miracle molecule. That's great, but we're going to have some leftovers also. And the leftovers are water and carbon dioxide. So we exhale carbon dioxide, we lose some water. And that is a really quick summary of what cellular respiration is. It's the making of ATP from one molecule of glucose. Where'd you get that molecule of glucose? Well, you ate some carbohydrate, you chopped it up into the little pieces, and now you have that molecule of glucose to use for manufacturing of ATP. None of this, I told you, can happen without enzymes. So they could happen in time, but it would take you 4 months to digest something. It would take you 6 months to move a certain muscle, so that won't work. So we have these enzymes that attach onto that reaction and speed it up and make it so much faster. So I want you to know the general name for groups of enzymes. If it's an enzyme that will break down proteins, it's called a protease. Notice the end here, ASE. If it's an enzyme that you have that you're breaking down fats, it's going to be called a lipase. And an enzyme that's breaking down carbohydrates is amylase. You have amylase in your mouth, so as soon as you put a piece of chocolate in or any carbohydrate, you already begin the digestion of some of these large molecules so that you can release the 1-glucose, so that you can make ATP, so that you can have energy to function. You will be reading more about enzymes in your book. They're never part of the reaction. They're actually sort of like a cab. They come, help, and leave. They come, help, and leave, right? An Uber. So they're not really part of what that reaction is. And there's two names that are referred to there: the substrate, which are the molecules that are reacting, and then some sort of product. So in this picture here, you have a substrate. Let's say it's a carbohydrate that you need to break down to use one individual glucose. So here it's just showing them as two. The enzyme, it's going to dock on the enzyme, and then the enzyme goes on its own way, and you have two separate products: substrate, products. Enzymes are really fussy. They can be destroyed very easily. Just the classic frying of an egg is a denaturing of a protein, meaning that you've damaged it. So the shape, because enzymes are all proteins, and proteins are sensitive. So when you fry an egg, it looks transparent, right? You have the yellow yolk and then the transparent white, and you cook it, you change the shape, it even changes color. So it means that you have rendered that protein sort of useless. It doesn't work anymore. Not that the component parts can't be a good part of your food, and they are. So fried egg is a denatured protein, which is a broken down protein or enzyme. Enzymes do the same. They're sensitive to pH and they're sensitive to heat. All right, I'm going to cut that mini lecture here. Please see the other videos. Please read your section in the book. And I'll see you for another mini lecture pretty soon. Bye.